Hello and welcome to the 20-minute wrap-up, and today we're going to be covering photosynthesis, which is over there. And basically what we're looking at is photosynthesis, the process that converts solar energy to chemical energy. And this affects almost all things in the world. Again, these are organisms that we call autotrophs, and they are the producers of our biosphere. Again, they make all these organic molecules out of basically water and carbon dioxide, and that's very important. We'll be talking about how that happens. This involves light, the visible wavelengths of light, as we can see here, and green things. Not only plants benefit from this, but animals as well. Again, those would be the consumers, and there are different types of organisms that photosynthesize. We tend to think of the plants, but algae, single-celled organisms, and there are things called blue sulfur bacteria that all can photosynthesize. Again, heterotrophs obtain their oxygen and their organic materials from the primary producers. Why is this important? We can also look at our fossil fuels, which are basically stores of sunlight energy that have been stored in the ground for eons and eons. And this picture here is just showing that, again, we are using photo energy to make energy in our modern age. Bacteria and chloroplasts are similar. The green from plants comes from chlorophyll, which is the green pigment in chloroplast. Carbon dioxide enters, oxygen exits the leaf through a microscopic pores called stomata, and that the chloroplasts are mainly found in a type of tissue called the mesophyll. The chlorophyll is in the membranes of the thylakoids. Okay, and it's stacked in columns called grana, and chloroplasts also contain stroma, and we'll talk about these parts here. So again, you need to know what all of these parts are. You need to know what a chloroplast looks like. The stroma is that material inside here. The granum are these membranes, and the thylakoid is an individual membrane, and the thylakoid space is on the inside. So it's very important that we know what these are. Again, photosynthesis can be summarized as carbon dioxide plus water plus light energy gives us glucose and oxygen. And a lot of times people give us a net and they remove this, but it's critical to know that water is involved in both of these. Again, this picture here shows you where the water is involved with, and that the oxygen that we get, that we breathe, actually comes from water that's split by these green algae. The oxygen in the water molecule actually came from the CO2. The hydrogen came from water, and we'll talk about that a little bit more. Again, in this photosynthesis, we're looking at a redox process in which the water is oxidized and the carbon dioxide is reduced. That is, we are adding electrons and hydrogens to the carbon dioxide. And so here we have that carbon dioxide, which becomes reduced. The water, which has hydrogen ions, loses those hydrogen ions to become oxygen. Photosynthesis consists of light reactions and the Kelvin cycle, which is the synthesis part of the photosynthesis. And the light reactions that occur in the thylakoids, we see where we split water, we release oxygen from the water, we reduce NADP, which is similar to NAD to NADPH, and we generate ATP from ADP by a process that we call photophosphorylation. The Kelvin cycle, which occurs in the stroma, forms sugar from carbon dioxide, and by using the ATP and NADPH that's made during the light reaction. And so what we see here, we see an input of light, we see an input of oxygen, NADP, ADP, and we get out ATP and NADPH. This is the light reaction. The dark reaction, or the Kelvin cycle, is where we see an input of carbon dioxide. We see a cycle where we basically regenerate the original molecules that we have. And this produces, in the simplest terms, CH2O, a sugar. And we'll talk about what that sugar is. Chloroplasts are solar-powered chemical factories. Their thylakoids transform light energy into chemical energy of ATP and NADPH. Okay, and so again, these are regenerated from other forms. Again, you need to know what life is. Not what life is, what light is. Visible light, we see here, occurs from 380 nanometers to 750 nanometers. Green is somewhere around 525 to 550. Again, we need to know this. Pigments are substances that absorb visible light. Different pigments can absorb different wavelengths. Wavelengths are not absorbed, are reflected or transmitted. Leaves appear green because chlorophyll reflects and transmits the green light. So it's very important that we know that. So here again, here's a chloroplast, and these chloroplasts are green because the photosynthetic pigment does not absorb green light. You should understand what a spectrophotometer is, and here's a spectrophotometer where we can shine specific wavelengths of light through a solution and measure how much light gets through. An absorbance spectrum is a graph plotted through a pigment's light absorption versus wavelength. The absorption spectrum of chlorophyll A suggests that violet and blue and red light work the best for photosynthesis. An action spectrum profile of the relative effectiveness of different wavelengths of radiation is driving a process. And so here, this is the action spectrum. This is the, and I'm not going to get into it right now, but the Eagleman's experiment, and here's the absorption spectrum. But basically, we see that the vast majority of activity occurs in these areas here, and that the least amount of activity and least amount of absorbance occurs in this area. And this is chlorophyll, and again, the difference between chlorophyll A and chlorophyll B. This is very, it's similar a little bit in a way to the phospholipids, in that we have a reactive head and we have hydrophobic tail. Again, this allows this organism to kind of hang out, and it looks something a lot like this. And what we see is it's like excitation of chlorophyll. When a pigment is absorbing light, it goes from a ground state to excited state, which is unstable. When excited electrons fall back to the ground state, photons are given off in an afterglow called fluorescence. And so, again, if we were to separate chlorophyll and charge it up, you would see this fluorescence. Okay. Again, a photosystem consists of a reaction center complex, a type of protein complex surrounded by light harvesting complex. The light harvesting complexes funnel the energy of the photons into the reaction center. The primary electron acceptor is the reaction center accepts an excited electron from chlorophyll A. Solar power transfer an electron from a chlorophyll molecule to the primary electron acceptor is the first step in light reactions. Okay. And so what we see here in this photosynthesis system, we have light harvesting complex, the light center stroma, and a special pair of chlorophyll A molecules is the thylakoid inner space. And this is a graphic showing here this interaction of those. But basically what's going on is we are exciting electrons, and this chlorophyll A donates an electron to the primary electron acceptor. There are two types of photosystems. Photosystem 2, which functions first and is the best of absorbing light at 680 nanometers, and reaction chlorophyll A of PS2 is at 680. Photosystem 1 is best absorbing at wavelengths at 700 nanometers. Again, 1 was discovered before 2. And what we're going to see is that there are a linear and cyclic flow of electrons. And there are 8 steps in the linear electron flow. In step number 1, a photon hits a pigment, and its energy is passed among pigment molecules until it excites P680. An excited electron from P680 is transferred to the primary electron acceptor. We call it P680+. Okay, this occurs in this region here. Okay, here's the pigment molecules. Those pigment, the light excites electrons that are given to P680, giving us P680 plus. Again, how this happens is that we strip electrons from water, and we release basically oxygen. The primary electron acceptor then transfers through redox reactions its electrons into a complex. At the second side of this complex here, we see electrons. The light energy from the light is used to further excite these electrons to a higher state. At this higher state, then we can use the electron transport chain to basically reduce NADP. The water is split by enzymes, and electrons are transferred from the hydrogen ions to the P680 plus, thus reducing it to P680. The P680 plus is the strongest known biological oxidizing agent. That's why it can work on water. Oxygen is released as a byproduct of the reaction. Each electron falls down an electron transport chain from the primary electron acceptor of PS2 to PS1, photosystem 1. Energy released by the fall drives the creation of the proton grain across the thylakoid membrane. Diffusion of hydrogen ions across the membrane drives ATP synthase. In PS1, like PS2, transfer light energy excites the P700, which loses an electron to an electron acceptor. The P700 plus, this missing electron, accepts an electron passed down from PS2 via the electron transport chain. Each electron falls down an electron transport chain from the primary electron acceptor of PS1 to the protein ferrodioxin. The electrons are then transferred to NADP and reduced to NADPH. The electrons of NADPH are available for reactions in the Kelvin cycle, and this process also removes hydrogen from the stoma. The energy change of an electron during linear flow through the light reaction can be shown in mechanical energy. And so in photosystem 2, what you're doing is you're increasing the energy of the electron. As this energy goes down, we're using it to make ATP. And then in photosystem 1, that energy is lifted a little bit higher, even greater than it was before, and we're using that to make NADPH. The cyclic electron flow uses not only photosystem 1 and produces ATP, but not NADPH, uses only photosystem 1. Cyclic electron flow generates surplus ATP to satisfy the higher chemical demand of the Kelvin cycle. And so in cyclic cytochrome, photosystem 1 excites an electron and continues to excite the electron, but again, in this electron, instead of going down and making NADPH reductase, it goes into the cytochrome complex and is used to make ATP. Again, some organisms, such as purple sulfobacteria, have photosystem 1, but not photosystem 2. They make lots of ATP. Cyclic electron flow is thought to have evolved before linear electron flow. Cyclic electron flow may protect cells from light-induced damage. Chloroplasts and mitochondria generate ATP by chemiosmosis, but they use different sources of energy. Mitochondria transfer chemical energy from food to ATP. Chloroplasts transform light energy into chemical energy of ATP. The spatial organization of chemiosmosis differs between chloroplast and mitochondria, but also shows similarity. In mitochondria, proteins are pumped to the intermediate intermembrane space and drive ATP synthase as they diffuse back into the mitochondrial matrix. In chloroplast, protons are pumped into the thylakoid space and drive ATP synthesis as they diffuse back into the stroma. And so this is showing you where the high concentration is, and then as they move down that concentration gradient, they make ATP. ATP and NADPH are produced on the side facing the stroma where the Kelvin cycle takes place. In summary, light reactions generate ATP and increase the potential energy of electrons by moving them from water to NADPH. Okay, and so this graphic here is showing exactly that. The Calvin cycle, like the citric acid cycle, regenerates its starting material after molecules enter and leave the cycle. The cycle builds sugar from smaller molecules by using ATP and reducing power of electrons carried by NADPH. The carbon enters the cycle as carbon dioxide and leaves the sugar named glyceraldehyde 3-phosphate. We saw this before. For net synthesis of one glyceraldehyde 3-phosphate, the cycle must take place 3 times, fixing 3 molecules of carbon dioxide. The Calvin cycle has 3 phases: carbon fixation, which is catalyzed by an enzyme called rubisco, reduction, regeneration of the carbon dioxide acceptor, rubisco bisphosphate. And so here's the ribulose bisphosphate, rubisco. So we input 3 at a time. So we have 3 of these guys over here. We have the short-learned intermediate. This produces 6,3-phosphoglycerate. Again, we put in 6 ATP. We get 6,1,3-bisphosphoglycerate. We put 6 NADPHs in it. We then have 6 glyceraldehyde 3-phosphates. One of these glyceraldehyde 3-phosphates then can be siphoned off, and the other glyceraldehyde 3-phosphates then are regenerated, combined with one another in order to form, instead of 5 3-carbon molecules, we get 3 5-carbon molecules. Okay. Again, dehydration is a problem for plants, sometimes requiring trade-offs with other metabolic processes, especially photosynthesis. On hot, dry days, the plants close their stroma, which conserves water but limits photosynthesis because you can't have it occurring without carbon dioxide. The closing of the stroma reduces access to carbon dioxide and causes oxygen buildup. These conditions favor a similarly wasteful process called photorespiration, which go in the opposite directions. In most plants, what we call C3 plants, initial fixation of the CO2 via rubisco forms a 3-carbon compound. In photorespiration, rubisco adds oxygen instead of CO2 in the Calvin cycle. Photorespiration consumes oxygen and organic fuel and releases CO2 without producing ATP or sugar. Photorespiration may be an evolutionary record called because rubisco first developed at a time when the atmosphere had far less oxygen and more carbon dioxide. Photorespiration limits damaging products of light reactions that build up in the absence of the Calvin cycle. In many plants, photorespiration is a problem because hot and dry, it can drain as much as 50% of the carbon fixed by the Calvin cycle. C4 plants minimize the loss of photorespiration by incorporating carbon dioxide into 4-carbon compounds in the mesophyll cells. This step requires the enzyme PEP carboxylase. PEP carboxylase has a higher affinity for carbon dioxide than rubisco does. It can fix CO2 even when the CO2 concentrations are low. These 4-carbon compounds are exported to the bundle sheath cells where they release carbon dioxide that can then be used in the cell cycle. So let's look at sugar production in C4 plants occurring in a 3-step process. 1. Production of 4-carbon precursors to catalyze the enzyme PEP carboxylase in the mesophyll cells. PEP carboxylase has a higher affinity for carbon dioxide than rubisco does. These 4-carbon compounds are exported to bundle sheath cells. Within the bundle sheath cells, they release carbon dioxide that is used in the carbon cycle. Okay. We also have a process called crullacine acid metabolism, CAM, to fix carbon. CAM plants open their stomata at night, incorporating CO2 into organic acids. The stomata close during the day, and CO2 is released from organic acids used in the Calvin cycle. Importance of photosynthesis, basically a review. The energy entering into chloroplast as sunlight gets stored as chemical energy in organic compounds. Sugar made by the chloroplast supplies chemical energy in carbon skeletons to synthesize organic molecules of cells. Plants store excess sugar as starch in structures such as roots, tubers, seeds, and fruits. In addition to food production, photosynthesis produces oxygen